Hello, welcome to part 2. I have to add in an edit and an apology from part 1. I've been writing and saying allotropic. I don't know why, it's just obviously a word that made more sense to me. And it's actually, if you look in your book, you'll see it's allopatric. Okay, so ALLOPATRAC. So I apologise, an edit, a rewrite. Okay, please go back through, and anywhere you've written allotropic, because that's what you've heard me say, please change it to allopatric. Okay, apologies. All right, so we're now going to keep moving on and having a look at some other kind of different types of speciation and mechanisms that drive speciation. So you've got our next kind of example that we need to talk about is sympatric speciation. And this is where you have populations of species living in the same place that become reproductively isolated either by mechanical, behavioural or seasonal changes. So our first example, our allopatric speciation, was a result of geographical and physical isolation. Okay, whereas this sympatric is because of mechanical, behavioural or seasonal, isolating mechanisms. In this model, some gene flow continues, which is really different from our allopatric speciation model. Okay, so in allopatric, because they're physically separated, they're geographically separated, you don't have gene flow. Remember, gene flow is the movement of either the whole organism or the sort of reproductive materials from kind of one population to another. All right, so it's something that doesn't occur in your allopatric version. Okay, but in sympatric it does. And so as a result of this, your sympatric species are actually closely related and they kind of occupy overlapping ranges. Now, kind of unhelpfully for you guys in terms of getting an idea with an example of what this could mean, scientists are actually reluctant to classify examples of sympatric speciation. So unlike our allopatric, where I could kind of say, think of marsupials, think of Darwin's finches, scientists are actually reluctant to give you an example of this. I guess because it's kind of hard to classify, I suppose. So for now, it's perfectly acceptable just to kind of have this definition in your head of what it is and how it is different from our allopatric version that we looked at. All right, so now we need to have a look at something called population bottlenecks. Now, as you guys will know from when we were doing our Harvey Weinberg work, that you need a large population to maintain a large and diverse gene pool. If the size of a population is greatly reduced, so is the gene pool and the allele frequencies. It's worth noting that even within a large population, your allele frequencies can be affected by factors such as mutation and non-random mating. Again, these are things that we spoke about in last lesson. We were looking at our Harvey Weinberg equilibrium equation. Okay, so you need those large populations to maintain large and diverse gene pool, and you kind of avoid reducing that and avoid reducing your allele frequencies. So hopefully it kind of feeds on from that and makes sense that if you have a dramatic reduction of your population size, you're going to have your reduction in gene pool. Okay, and your dramatic reduction of population size can be caused by loads of different things, environmental disasters, a new disease, hunting, either that's by humans or kind of a new really successful predator, or habitat destruction. And this dramatic reduction in size kind of forced upon a population, so caused by things that are outside of their control, is called your population bottleneck. And so this severe decrease in the gene pool, many genetic variants are going to be lost, and in almost all cases your genetic diversity is greatly reduced. So less organisms, smaller gene pool, a loss of gene variants, a reduction in your genetic diversity. And so your remaining small population, so what you're left with after your population bottleneck, is vulnerable to the complete loss of some alleles, and a single mutation or a new individual can have a bigger effect than usual. Okay, so think about, we've spoken about this previously, if you've only got 10 organisms in a population, that mutation, and one of them has a mutation, it's far more likely that mutation is going to be passed on and it will increase in frequency because there's only a smaller number of organisms to, kind of mate with each other. Larger populations, the impact of that mutation can kind of be minimised. So once you've had this population bottleneck occurring, our population, as we said, is kind of really vulnerable to some alleles being completely lost, and your mutations in new individuals can have a bigger effect than usual. So as your population then starts to recover, starts to increase in numbers again after this kind of devastating catastrophic event, the population can become so different from the original one because of our allele frequencies that actually, it's so genetically different and that it has to become a new species. So population bottlenecks can be another kind of driving force behind speciation. All right, so the last thing I need to do with you is something called the founder effect. So if you give me 1 second, sorry, I'm just going to get the pieces of paper ready. Okay, so the founder effect is similar to your bottleneck population in the terms that you get a loss of genetic variation, okay? But in your bottleneck population or population bottleneck, the reduction is forced upon them, okay? So it's an event that happens. The organisms aren't actively making that choice. Whereas in a founder effect, it's voluntary. So what happens is a small number of individuals leave the main population and set up a separate new population. They're not forced out by an event. They're not forced out by, human interaction or a new predator or anything like that. It's done on a completely voluntary basis. And that's your difference between your founder effect and your population bottleneck, okay? Now, the alleles carried by the individuals who leave the main population, so your alleles carried by this founder group, are unlikely to include all of the possible alleles. It's highly unlikely, in your smaller group that you're going to have all the genetic variations covered. And it's highly unlikely that they're going to be at the same frequency as the original population. If you've got your original population is 500 and an allele is present in 10%, it's highly unlikely that if 10 of those organisms wander off to create a new population, that allele frequency is still going to be the same. And so what this means is that any unusual genes in the founder population may become more frequent as the population grows. And so in the same way as your bottleneck can occur, what that means is you can end up with kind of a new species as well because of this founder effect. The way or an example of this that they give you in the textbook is about the Amish population. So in the Amish population, or the Amish group of people, a genetic disease is considerably more likely than in the normal population. So your genetic disease is Ellis-Van Creveld syndrome, and in the normal population, or the general population, I should say, it only affects about 1 in 200,000 births, whereas in the Amish population, it's kind of been recorded as 43 out of 8,000. So because of this founder effect, because that mutation was present in one of the founders of the Amish community, and because they kind of were only able to reproduce within their community, it meant that that mutated allele became far, far more common. And so obviously the phenotype is more common in their population. Now, we wouldn't class Amish people as a new species at all. That's just an example of how, the unusual gene can become more frequent. But, if it were to carry on to a particular point, then it is possible that your founder effect could result in an entirely new species. All right, so that's everything I need to talk to you about. Please let me know if you've got any questions.